Detector might be very special.
Okay. Okay, I think it's time to get started. I'm going to just start and do a quick introduction. And I think if people can just, I think everybody, all right, so we're quiet here. Can everybody in the online hear us? I got it, it's a good here. All right, let's see. Oh, good, so everything's working, except we have in here in our room a little thing in the middle of our screen, but we'll work through that. It's not on, no. So, and it comes and goes. So welcome to the second seminar in the Northeast Regional Environmental Acoustics. Today, before I introduce our speaker, I just wanted to give you a preview of what's coming next. So on November 14th, Andoni Lavery from the Woods Hole Oceanographic Institution will be giving a talk. She has received a prestigious Monk Award that she will be giving the award plenary talk at the Ocean Sciences meeting later in December. Is that correct? And she will be giving us a preview of that talk in November for people who aren't going to be able to make it to the Ocean Sciences award lecture. So we'll be very fortunate to hear that. I also wanted to let everybody know that we now have a working, in progress website. And let me see if I can get to that. Oh, look at that, it's going, great. The one issue we're having right now is when you come to the homepage, there's nothing on it. So we have to fix that. But if you go to the tabs, they actually do work. So here's where you can get a bunch of information, and it actually loads slow here right now. There we go. So you go about, you talk. I also wanted to bring up the, so these are the goals of our environmental workshop. The working group leaders, if you have any questions or comments that you want to get across about the working group and how things are going for the seminars, feel free to contact any one of the 6 people on the steering committee. Oops, I can get this down. So they're all listed here. Activities is the last tab I will show. Shows our upcoming events. Information. Yeah?
Says your screen sharing is paused. You sit on your laptop.
Oh, your screen sharing is paused. I don't
share.
Resume share.
I don't know, it was still on your screen. That's what I was looking for.
So I don't know why that's not working. I'm just trying to get to, oh, here we go. Everybody see, oh, it's paused again.
Oh, that's pauses.
So the hyperlink pauses it for some reason. But I encourage you to go to the website, and there you'll see information about the symposium on March 20th. And at the bottom of the activities page and on the resources page, you'll see linked seminars to past ones. So if you want to view the seminars from last month or as they stack up, you can go to that page and direct it to your students or anybody who might have missed it. With that, let me just make sure. Whoop.
So Zoom allows you to share an application or your desktop. You probably
yeah. But right now, we're not having any issues with that. It's all showing up right. And so I'm going to introduce Dr. Dan Howard. He's a professor here in the Department of Biological Sciences. He's not underwater, and it's really great. After last month, and next month we'll be underwater. We're trying to alternate with terrestrial. So he'll be talking about terrestrial environmental acoustics today. I will let you take over.
All right. We can keep this working, and maybe you can gauge from our online audience if they can hear me okay. And because I tend to wander when I give a talk, and I'll try not to do that, so I stay close to the mic. So I do want to apologize just a wee bit, because I'm not going to talk a lot about marine acoustics. I purposely found a couple of examples to use simply to be inclusive in what I try to do. But I'm a terrestrial biologist, so I study animal behavior. My training is kind of in bioacoustics, so I'm interested in how animals communicate. And so my PhD was studying acoustic communicating insect species, and so I'll talk a little more about that system a bit later. But what I find myself, among other things, really interested in now is how animals make high-stakes decisions in very complex acoustic environments. In particular, you guys have all heard about the idea of the cocktail party problem, right? I mean, we were dealing with it when everybody came in here at once and everybody was talking, and maybe the person that you were sitting next to, you were trying to have a conversation with, right? And you got some masking stuff going on. We had an interesting example of a multimodal masking agent, and for a minute we had something, I'm not sure it won't come back at some point, but we had something flashing on the screen here that could have acted as maybe a masking agent in terms of the information that's on the screen. So what I find myself really interested in now is really addressing kind of how the cocktail party problem is resolved in a biological, using biological solutions in a multimodal sensory context. And that's kind of a different way of thinking about how systems have evolved to address the masking problem that's intrinsic to what we typically call the cocktail party problem. So I'll come back a little bit later and revisit the cocktail party problem, the whole issue of multimodal sensory context, and then a little bit at the very end on some of the things that I'm working on, my students are working on addressing those sorts of questions. But what I want to mention from the beginning is when we were talking about the seminar series, Jen had asked about, well, we need a bioacoustics talk that's kind of experimental bioacoustics. And so I raised my hand thinking, okay, I could probably cover that in a terrestrial context. But as I began to think about that, I felt like we need to start by kind of defining what that means, or at least what it means to me, so that when we're talking about experimental bioacoustics and I'm giving you examples of that, this is kind of where I'm thinking of this. So last month, we heard a talk that was looking at the bioacoustics of a marine mammal, and we learned a lot. I learned a lot about signal structure in these organisms, how they differ between the two focal species, almost in the context of a syntax, like we would think about that in the context of bird communication or even human language. And so when I'm describing what bioacoustics is to somebody who's not necessarily a bioacoustician, what I generally say, it's an innovative science that fuses biology and acoustics. So you got both the biological and the physical sciences represented in that endeavor, and it's really looking at questions related to sound production, dispersion, and I'll define that in a minute, and then reception of that sound in animals.
I can't hear you.
Okay, so I'll move back over towards the mic a little bit. Like I'm getting some signals that maybe I'm talking away from the mic. So really, it's just kind of looking at how sound is produced, how it transmits to the environment, and how it's received in animals. We'll revisit that definition. But when I was thinking about experimental bioacoustics or bioacoustics in general, we really need to kind of use this as our frame of reference, right? So when we
Use the mouse, no one can see.
Oh, okay, yep. So that's right. Only you in the room can see my green pointer. So the model that we typically use in bioacoustics is this model, and this is the classic sender-receiver paradigm, where a sender is producing sound somehow, and we can also apply this to other communication modalities other than just sound, but we're talking about sound today. So you have a sender that's producing a sound. Last month we talked about leopard seals. What was the other species of seal that we learned about last month?
Humpback what?
Humpback whale, I guess, was the other system we talked about. So you've got a sender that's using some sort of biomechanical mechanism to generate a vibration, modifying it somehow, coupling it to the transmission environment. Water last week we looked at, propagating it through that medium, and then a receiver decoupling that sound, modifying it to some degree, and then classifying it through some process of perception. So when I think about experimental bioacoustics, the twist I add to the traditional definition of just bioacoustics is that it's the experimental manipulation of sound, or its dispersion in the transmission medium, or its reception in order to understand the ecology or evolution of animal life history and behavior. So that's kind of my working definition for experimental bioacoustics. Bioacoustics in general is a very old field, right? And it emerged from just direct observation of sound production, recording of sound production. So when I think of experimental bioacoustics, I think about ways that we actually manipulate that process, not just record and document sounds. All right, that's fair. That's kind of my working definition for what I'm thinking. How I'm thinking about this. Likely a good candidate for the founder of what we call experimental bioacoustics is Ivan Regin, who worked really kind of at the turn of the century through the 20s. And his work was experimental in that he was interested in how crickets communicated. And so specifically, he had a big tank in his lab they worked with. And not only did he observe and record, which in the 20s was a very rudimentary mechanism for doing so, but not only did he observe and record the sound, but then he actually manipulated the sound. He did some really simple playbacks through a speaker. He did some modification of the sound producing structures in the cricket. So he actually was kind of considered the founding, at least among terrestrial biologists, the founder of modern bioacoustics, and especially experimental bioacoustics. In the years after that, so he was really active in the 20s and 30s, and the years after that, there's been a lot of other bioacoustics folks that have contributed to terrestrial bioacoustics. I think a lot of insects of North America library, which is kind of the analog in invertebrates to the Macaulay Library for birds. He's emeritus at University of Florida. Dick Alexander is active about the same time. He was really interested in sound and how it was a driver in sexual selection and evolution of traits. And then there's lots of others I could mention. This is not an exhaustive list, but Glenn Morris is also University of Toronto, did a lot of early work in experimental work, doing active playbacks using vibrational signals and katydids. Most of us are familiar with Jack Bradbury's work with birds, seminal bioacoustician, as is Ron Hoy, who works in physiology. I mentioned a couple of other people on here that I just want to throw out there that you may not have heard about in bioacoustics, and one is Andrei Kokol, who's a Slovenian who works in vibrational communication. So he studies sound or bioacoustics through substrate-borne signaling, and his work is led in a lot of different directions. A lot of his students now are doing experimental work where they even do playbacks of vibration to control, for example, mask the signals of certain pest species of grape leafhoppers and control, kind of in an applied context. And then I throw out Monica Gagliano at the University of Western Australia as an example, simply because my definition is focused on animals, and in the last few years there's been more and more talk about bioacoustics in plants. And so that throws another wrinkle into that. We don't have any speakers, I think, in our seminar series talking about bioacoustics in plants. But Monica has authored a series of papers, I think from 13 through 15, really arguing that plants have many of the same traits for, in some cases, producing sound and responding to sound. So add that to your thing to think about is, should we include actually plants in bioacoustics as well? And I will also, as a caveat here, say I'm going to probably pose more questions than I'm going to answer in my talk today. So thinking about plants as a cohort member in bioacoustics is one of those things I want you to think about. All right, because a lot of what we talk about, as we did last month, and what I'm going to talk about today is really kind of focused on acoustics in the context of animal communication, we have to take a step back and think about another big player, who is Nico Tinbergen. And most of you probably remember this fellow. He worked with two others. He earned the 1973 Nobel Prize in Physiology or Medicine for standardizing kind of the way we think about studying animal behavior. And the reason why I put him up here is because this is the way typically I think about it. A lot of those who study animal behavior think about studies in bioacoustics. We focus in on either looking at the actual mechanism or the causation for how sound is produced or received, how it changes over the development of an organism, its ontogeny, its function. In other words, what if a bird is calling a song, what is its adaptive value for putting all that effort into a signal? And then the evolution of a particular acoustic trait and how it might map out across the phylogeny. So that's the framework for how typically those of us who study animal behavior in the context of bioacoustics think about how we frame our questions. And you'll see that most of what I talk about today when I touch on my own work is focused more on causation, mechanism, and adaptation. Those two areas are most of what I think I spend most of my work with. So we've talked about bioacoustics, experimental bioacoustics, kind of the framework that we use for that. Now let's circle back to the other part of my title, which is a cocktail party problem. We mentioned that a little bit. This was for this idea of the cocktail party problem. And for some of you, you're thinking right now, the only problem I have at a cocktail party is how quickly I can get a cocktail, right? But the idea in the cocktail party problem is the issue of masking, right? When you have a lot of voices, when you have a lot of sound being produced by multiple senders, then oftentimes, though, the information in that signal might get masked by another source of sound. And so Cherry in 1953 authored a paper that kind of formalized this idea of the cocktail party problem and the issue of masking in animal communication and acoustic communication. Since then, if you did a search for cocktail party problem in Google Scholar, there would be hundreds of papers since 1953 that have come out touching on this. And so I cited a couple of them here. I tend to think of the cocktail party. I tend to be interested in how organisms solve the cocktail party problem in the context of social living organisms, because those are the ones that are typically challenged with that. All right, so it's an idea of signal masking, and signal being the sound that's produced. If we think about the humpback whales or the leopard seal, those guys were producing sound. There's a potential for some sort of other sound to actually mask that information in the transmission media. So think about our model, think about where masking is likely going to take place, and it's generally in the propagation phase of this sender-receiver dyad. Okay. But this problem has been around for much longer than since 1953, right? So organisms that produce sound have been dealing with it for a very long time. If you think about certain systems, animal systems, and I don't have anything into all in here about plants, but animal systems in which there are multiple senders and receivers found in a single place, the opportunity for the cocktail party problem is obviously highlighted, right? And so a lot of our work with respect to understanding the neural mechanisms of this problem, which it really is kind of a receiver problem, has been focused on systems where you have multiple signalers interacting with each other at the same time. I'm giving you all examples of terrestrial systems, but in marine settings, I think of like snapping shrimp, I guess, or pistol shrimp that are making a huge amount of noise in the context of some shallow marine settings. Off the coast of New Zealand, there are sea urchins that are constantly feeding it a couple of times a day that produce just a cacophony of noise. It's biotic noise, but it actually can act as a masking agent in the context of this sender-receiver model. Okay, so let's talk about a few solutions that we've discovered in organisms that have been dealing with this problem for the last, in the case of invertebrates, 180 to 200 million years in the Incifera. But typically the simplest solution to the cocktail party problem is to just, if you're the sender, just be louder, right? So how many of you, I noticed in a classroom, if I try to stimulate conversation and there's more than about a dozen students in there, at some point that amplitude level just gets higher and higher. And oftentimes we use it as a proxy for engagement, right? It's really loud, it's boisterous in the classroom. Really all that's happening is the Lombard effect, right? Everybody is trying to make their voice loud enough that the receiver can hear it. So that's one simple behavioral solution to that. There's also other ways that senders can deal with, behaviorally can deal with the cocktail party problem. Is that working okay? Oh, okay, all right. Oops, let me pull this around this way. Is that working?
There we go.
Is that better?
Okay,
thanks for managing the mic here. You can increase the amount of acoustic energy that you're producing, not the amplitude, but maybe the number of signals that you're producing. That's another sender behavioral solution. You can also just be quiet, right, and not produce sound until the background noise declines a little bit. That's another simple plastic sort of behavioral response to noise or the cocktail party problem. And also, there's some evidence that some organisms will actually seek out habitats that have better transmission properties, lower levels of masking noise. So those are all sender solutions. Those are behavioral or ecological solutions for a sender to overcome the cocktail party problem. There are also adaptive solutions that senders can evolve in the context of constantly being challenged with a cocktail party noise sort of problem. We can think of this in the context of a couple hypotheses that behavioral ecologists and bioacoustics cite when they're testing models for how organisms might deal with a noisy environment. One is the acoustic adaptation hypothesis, which basically simply states that signals become adapted, spectrally, temporally adapted to the noise level in the environment through which the sender would be sending those. The other is that there may be niches, acoustic niches, right? So you think about the idea of a niche in ecology. Signals can also evolve to fill particular niches. Maybe they're spatial niches or spectral niches or temporal partitioning, time of day, when you, as a sender, choose to produce a signal, right? So all of these things are more adaptive solutions to that cocktail party problem. However, the problem of noise and masking noise is not a sender problem, right? It's a receiver problem. So if you're in a noisy environment, really the biggest challenge, the biggest fitness cost to organisms is on the receiver side. So most of the receiver solutions to the cocktail party problem are adaptive solutions. And so we can think about several ways that organisms might evolve to overcome the challenge imposed with a noisy environment. One of those is frequency tuning. So here's an example of a frequency tuning diagram off to the right, where maybe if you're in a very spectrally complex environment, the band of sound that their species is producing sound. And we see this a lot in neotropical katydids, where there might be multiple species, and they partition out not just the signal from the sender standpoint, but the receiver's auditory system, sensory system, co-evolves to be almost niche segregated and narrow tuning. There's also the idea of spatial release from masking, which if you can separate yourself from the noise source to some degree, you might be able to increase the intensity of the actual sender's signal. There's gain control, which is another sensory adaptation for basically just paying attention to the loudest, potentially the loudest signal in your acoustic space. There's also novelty detection, which is another mechanism for potentially just coding maybe a particular part of a signal that will allow you to tune out some of the noise. Increasingly, what we're starting to see too, and these are all thinking about this in the context of noise being bad, right? Masking information resulting in receiver error. But sometimes what we've seen is that noise can have a benefit to receivers. And this is something we first kind of recognized in kind of neuroscience, functionally, or sensory biology, where we think of this idea of stochastic resonance, which auditory nerves will respond better if there is some small amount of background noise rather than absolute silence. And then the other is what we've seen in frogs especially, and we're starting to see this emerge in some studies with insects, is that if the noise is co-modulated, in other words, if it's biotic noise, it has some sort of temporal pattern to it that is similar to the actual signal. For example, if in some species of birds where the call song is particularly temporally stereotyped, if there are a lot of birds calling, that background biotic chorus noise, you might consider it, actually the sensory system, the receivers can tap into that to actually perform better with that, as long as the noise is co-modulated, and co-modulated in a way that's biologically relevant. We see that in certain species of frogs that mate in big, what we call scrambles. All right, so those are all behavioral and potentially some adaptive solutions to this cocktail party problem. And so when I think about this whole particular issue, all of the examples I've cited, and most of the literature, when you do that Google Scholar search and you see hundreds of papers on the cocktail party problem, almost all of them are looking at noise in an isomodal context. And so what isomodal, or some investigators refer to as unimodal, is referring to is when the noise is in the same sensory channel as the sensory reception modality. Does that make sense? So if you're listening to a conversation and somebody is sending an acoustic signal, then the noise is in that same sensory channel. That's isomodal. Most all the work that's been done is looking at the cocktail party problem in that context. Increasingly, though, and this is an example of an isomodal noise source, where the actual signal source here might be masked by an envelope of noise that covers up that same signal, right? So the idea is masking in one channel, signal in the same channel. That's the classic example of the cocktail party problem. Increasingly, so this is isomodal noise. Definition is it occurs in the same sensory channel as the detection and processing of that. In the last 5 years or so, what we started to try to understand is how the cocktail party problem is resolved when there are multiple channels on the sensory side that are engaging with noise. So one way we can think about this is in the context of cross-modal noise or cross-modal effects. And this would be where the vocal signal is received through one modality, for example, through the auditory system, but maybe the noise is in a different channel, maybe a visual stimulus, and how that system works on the sensory side and how it results in behavioral adaptations is something we're still trying to sort out. So that's an example of cross-modal noise. We can also think about multimodal noise, where maybe the noise itself, the cocktail party problem noise, is actually in two different channels, or you can even imagine maybe more than two channels, and the sensory system is engaged with that in a couple of different sensory modalities as well. For us, maybe vision and hearing at the same time, or maybe even olfactory noise could be introduced as well. So this is kind of where, in terms of the cocktail party problem, as bioacoustics professionals, we begin to think about how have biological solutions arisen in the context of these more complex challenges in animal systems. So we can think of some examples of these. Here's my effort to try to bring marine acoustics into this as well. Some isomodal effects, we can think about how, in the context of disturbance and interference of signals, we can think of how blue whales are disturbed by acoustic noise, whereas bats, I'm thinking of big brown bats, how their signals are actually interfered with, right? So we've got, and those are both noise sources in the same channel as the primary sensory modality. Here's an example of some cross-modal effects where we have acoustic noise actually disturbing visual signaling in squid and interfering with the processing of those visual signals in hermit crabs. So that's an example of a cross-modal effect, right? In multimodal, a couple of examples of multimodal noise effects in the cocktail party problem would be where light and noise pollution has an effect on male robin calling behavior, and where anthropogenic noise is traveling through air and also along the water surface in Tungera frogs influences their calling behavior. So can you see now the idea of multimodal, cross-modal, where you're actually interfacing with a couple of different sensory modalities, and the solution now becomes more complex, right? Because now you've got potentially noise masking in a couple of different ways, not just in the primary or isomodal channel, but also in secondary channels as well. So my interest dials back to what I mentioned at the very beginning of the talk. My interest is really how animals make really high stakes decisions. So mate choice, parental care, predator avoidance, those sorts of high stakes, life and death sort of high fitness cost decisions when they're faced with these sorts of noise, cocktail party problems across different sensory channels. Does that make sense? So this is where kind of in sensory biology and bioacoustics where neuroethologists are trying to sort out what are some of the biological rules for solving these problems. So, and again, we go back to the Tinbergian approach to this, is looking at what's the mechanism for organisms that might be solving this, how does this problem exist across developmental stages in organisms, and how these solutions might be adaptive. And then we haven't even got yet to the point of really sorting this out at the evolutionary level. All right, so some examples of some of the, so that's all background basically on what I'm interested in, and hopefully a lot of this is, the ideas of this are things you understand, have heard a little bit about. So my work really, I'm gonna give you some short examples of some work taking place in my lab, and then dial in on one last example where I've delved in a little bit deeper with a system that seems to be a very good model for studying the cocktail party problem and its solution in a multimodal context. So one system that I was fortunate enough to start working with as a postdoc is a group of insects found in New Zealand, and they're called giant weta. They're related to a cricket, so they look like a cricket except for Brooke has worked with them before. So Brooke has worked with giant weta. She's gone to New Zealand with us actually a couple of times. Yeah. So they're big crickets that are like this big, basically. And one of the interesting things about New Zealand having, and Sarah's in the back, smiling, grinning back there, because she's been to New Zealand as well and worked with these guys. So these guys basically, like all the other life in New Zealand, evolved in isolation for 80 million years. And as it turns out, the giant weta ancestors lacked wings, for the best we can tell from phylogenetic reconstruction. And so if you're a cricket, if you're a member of the cricket family, you know how they produce sound, right? How do crickets produce sound?
Stridulation.
Stridulation, right? So they rub body parts together, common way for invertebrates to produce sound. We make sounds sometimes the same way as well, inadvertently. So these guys don't have the architecture to do this, right? I'm not pointing fingers. So they lack the architecture. They lack the parse riding mechanism. What they do have is some pegs on the side of their legs. They can do a little bit of stridulation in an anti-predatorial context. But what's happened in the evolutionary history of giant weta, there's 11 extant species in New Zealand, is they've evolved to use substrate-borne sound. So they actually vibrate and produce a signal that is transmitted in whatever substrate the particular species tends to make its living in. So there are a few species that are ground-dwelling, like this particular species, Deinocritta rugosa. They produce a signal that gets transmitted to the substrate and gets received through subgenual organs, which are organs that are found in the legs of insects and that are the precursor organs for hearing in insects. And so they detect the vibration. This particular species seems to have a very simple repertoire. It seems to send just signals in an agonistic context. So they use vibration. There are some species that have a more complex repertoire, apparently, some of the arboreal species. So some of our work here is looking at how vibrational noise affects their communication system. And in this context, they actually hear auditory sound. They hear very well, but they communicate through a different channel. And so some of the questions we're looking at here, and Brooke and Sarah actually were involved in some experiments last year on this question, but how, for example, auditory noise or acoustic noise, airborne noise, would affect their behavior and their communication when they're actually insects. And she's looking at this in a different high-stakes context in parental care. So some of you know that in my lab and in Dr. Hall's lab, we use burying beetle models. And burying beetles are these little brightly colored beetles, orange and black. You can kind of see them down there in the lower right. These guys are notable for using biparental care in their life history. So they go find a little dead carcass, they'll bury it, they're producing sound during this whole process, during pair formation, courtship, burial, even during parental care. So they're producing sound. We don't really understand how they receive the sound. In fact, there's some evidence that maybe they don't receive the sound at all. They're using the substrate-borne vibration produced by that stridulation behavior to encode information. But Mia Phillips is studying how noise through that channel is affecting the parental care behavior. And that's another project that's taking place in my lab. I had a student who's now a master's student in my lab, Nicole Abadi, who as a postdoc was really interested in how this whole question of multimodal and cross-modal noise, how it affects circadian patterns. And so thinking about how organisms budget their time between resting, feeding, mating, fighting, all the things that all organisms do. She was interested in how exposure to airborne noise, exposure to substrate-borne noise, which is biologically relevant if you're a cockroach, right? So you've got those 6 legs, all of which have subgenual organs that can transduce vibration. And then how in combination those affect the behavior. So she had a prep set up where she was actually monitoring their behavior over 24 hours, over multiple days to kind of build an actogram for when cockroaches are active. As it turns out, we discovered these are Madagascar hissing cockroaches, so these are big rascals about this size. We learned that they're active at night, so that was a great discovery for us. Everyone who's ever rented a dodgy apartment, and we know when you turn the light on, cockroaches come out. So we discovered that Nicole discovered that these guys also were active primarily at night, but she also discovered that when she boils down and distilled their activity patterns, that when she subjected them to acoustic noise or vibrational noise or a combination of the two, it would actually change their activity pattern. So they would rebudget their time over the course, and these were over the course of several weeks. So this is kind of a new thing we're beginning to understand, is that exposure to multi-sensory, multi-channel forms of noise can actually cause animals to change basically the way their circadian patterns work. And so this is still a project that's ongoing that we need to collect some more data on. To show that I do have a little bit of interest in marine acoustics, although I'm not all that great at doing work in sound underwater, as it turns out. I had an undergraduate student who was really interested in, she had an interest in marine biology. She also was interested in invertebrates. So she read some papers in one of my classes about how the sea urchins in New Zealand would produce this really loud noise when they're feeding on parapython rocks. So she ran a project where she basically did noise playback, but the noise that she synthesized and played was actually a replicate of their feeding sound. And her idea was to test whether or not the sea urchins were actually, without lacking hearing organs, if they could transduce the vibration from the substrate and actually eavesdrop on that sound and, as a cue for maybe feeding behavior and conspecifics. So what she found was that it wasn't the case, right? When she did playbacks, they didn't exhibit any sort of phonotaxis towards the speaker, even though in this one image it kind of looks like that's the case where the sea urchin moved from the center towards the speaker, but in most cases that wasn't. But what she did discover is that within this fairly loud noise that we introduced into the tank, that it actually slowed down their overall vigility. So they were actually quite unwilling to move in the context of a lot of noise. And this is just a little undergrad project that I wanted to highlight to show that sometimes I do get my speakers wet once in a while. But this is what I want to talk a little bit more about, because this is the system that I think represents a fairly good model for studying this idea of sensory adaptations from multimodal, cross-modal noise, and how organisms over time evolve solutions to these sorts of problems. So during PhD, I started working with this particular species. It's called the prairie mole cricket, Grillotapa major. It's a fairly rare insect. It's only found in Missouri. There are other mole crickets in North America. Many of them are introduced species found in as turf grass pests down in the south. There's one other native species that's more of a solitary species, but this species is unique in that in the tallgrass prairie remnants and a little bit of habitat or ecosystem that they produce these really interesting little acoustic acoustic burrows, what we see right here. They gather in large numbers. There's a GIS map there of maybe 30, but sometimes there are as many as 100 or more. All of these males clustered kind of close together, all producing acoustic signals that are meant to do one thing. And I think we'll see if we can play this and hear it here. I don't know if this will work. I didn't test it in advance, but let's see. I can play it one more time. I don't know if it's gonna play for our audience here or not. Maybe they'll hear it, maybe they won't. But basically, all I can say about that is it sounds like a cricket, right? I mean, so the males do this, but it's very loud. This doesn't really, it doesn't give the full intense sensory experience of listening to 100 of these calling at around 100 dB from 20 centimeters. So they're very loud. Their acoustic burrow amplifies that sound, and they do this for only about an hour at dusk. And so right at dusk on the tallgrass prairie, as the sun is setting, all of these guys fire up. And so you can imagine that poses a bit of a cocktail party problem, right, if you're a female. So what I worked on in my PhD was really kind of understanding a little bit about their sound production. We learned a lot about what the sound looks like if we break it down in its small pieces, how they produce sound, like most crickets did, like Dr. Hall showed. They rub their wings together like this. They have a pars strident. Their call song is about 2 kilohertz. It's about 2.5 chirps per second, and it exists with about 17 to 20 syllables per pulse. So we know a lot about this sort of behavior. Males come out every evening. They tend and retune their burrows right before they begin calling. So it's almost like getting ready for the ninth party, right? Got to clean the pizza off the roof and all that sort of stuff. So they retune it. What else do we know? We know that part of what I learned in my PhD through some of my studies was that in the field or in the lab as well, that the dominant frequency of the male call song was correlated with size. So, and this is kind of what we see across systems, right? If you're bigger male, you produce a lower dominant frequency call. This was the case with these guys as well. We found a little bit about the ecology of how where the males like to display and produce their call songs on recently burned grasslands. They didn't have a preference for soil types, which we thought might be the case because they build these little resilient acoustic burrows. The burrow design was highly variable between males. So there seems to be like some sort of personality in terms of how the males produce their burrow. We also found that if you look at the little map here, that there were really, unlike other leks, like in sage grouse and prairie chicken, where there's like prime real estate in the middle of the lek, and all the females go to the middle, and the males that are in the center tend to have higher mating success. That wasn't the case with these guys at all. In fact, it seemed like the males around the periphery were. All right, so I'm interested in female mate choice. What are we missing here? Anything about females, right? So, like has been common in a lot of bioacoustic studies, everything we learn about a system typically initially is about the males. And part of it is males are simple to study. Males are simple, period, right? So it's easy to know what males are doing, why they're doing it, and how they're doing it. So that's really what I spent most of my PhD working on, is male behavior. But during all of this, what really became highlighted in my mind is what's going on with the females, right? They're really the ones who we should be studying, because here is the perfect model of the cocktail party problem. We could have up to 100 males producing 100 dB calls, all of which are mostly overlapping. How in the heck would a female, how did she locate a mate in this clearly cocktail party environment? How has their sensory systems adapted to extract information when you have all these males screaming at the top of their parse strident to come mate with me, right? That's what's happening. And the big problem is I didn't even know how to collect females. This is the problem. When I started working with this system, nobody had published it. Oh, by the way, the first acoustic work was actually done by that fellow Tom Walker I mentioned at the very beginning. This was a species, I didn't say this, species presumed extinct until 1989. And then around that time, Tom Walker and a agency fellow from Missouri, DNR, rediscovered them. And so he published a little bit of work on their bioacoustics. I followed suit along with Dr. Peggy Hill and describing all this wonderful stuff about the simple males. And then here we sat knowing nothing about really what are the more interesting parts of the life history, which is female behavior. How they make mating decisions, these high-stakes sorts of decisions. So the biggest problem for me as a postdoc, when I really wanted to get into the sensory side of this problem, was how the heck do we even find females? It's getting dark. We can't see. So luckily, if you were going to try to find females, how would you do this in this system? What's that?
Find the males.
Find the males, right? So actually what we did is we used males. Let me see if I can get this to work. Males as bait. And so basically, I'll probably cut the lights on it maybe. But anyway, maybe you'll get the sense of it in just a second. This is a female prairie mole cricket who has responded to a real calling male. So this is the male who's calling. But unlike most males who have their burrow open where the female would then go inside and they would start doing courtship. Instead, I did the very mean thing of putting mesh traps over the top of every male on these different leks. And so what I did is I used the males for bait. And so by this mechanism, I could actually collect the females and get to the more interesting. This was like a big thing for us to actually get our hands on females. We'll stop this here at this point. So that allowed us to actually use a trap like this. And this is an example of one particularly sexy male who in one night attracted something like 6 females. So clearly what we're interested in is what traits, what acoustic traits does a male have, and what sensory traits do the females have that allow them to wade through this cacophony of the cocktail party problem and actually find what would be an attractive male. All right, so a little bit more about this story as it plays out. First thing that I had to do was collect females. I say there was other people involved with this too. I had a student who was a PhD student at the time at the University of Toronto, Dr. Norman Lee, who's now faculty member at St. Olaf College. Dr. Kerry Hall was intimately involved with all of this, running around the prairie, avoiding bison and lightning strikes to do all this work. And a couple of my students, actually several of my students have been to the site, so they know what this looks like there. But what we really needed to do was collect the females that were responding to the male advertisement call, assess what they actually were perceiving, right? So this is really what I was interested in. What do the females actually hear? And not just what do they hear, but also what do they sense in terms of vibrational noise? Because what we've learned about males, what I didn't throw up there as well, is males seem to be using the vibrational component of their calls to space themselves. In other words, they weren't listening. In fact, their whole audit males, when they were calling, seemed to be turned off. You could go up and have a loud conversation around a calling male, and they would never pay any attention. But if you tap your foot on the ground, they'd stop calling. So they used the vibrational sensory modality to actually, for the most part, seemingly detect predators. So what we learned by doing some exercise, without going into all the details of doing how we do the extracellular neurophysiology in invertebrates, what we learned is that females have and males have very narrowly tuned sensory systems. So males call at 2,000 hertz, 2 kilohertz, and their tuning sensory system hearing is tuned narrowly for that. The other thing that we discovered from this when we were doing the physiology is they have a second peak out in the ultrasound. So I might ask you, why do you think they would have a second hearing peak in the ultrasound? Probably a bat detector, right? We haven't really tested that. I have done, spoiler alert, I've done some tests with females on the ground responding to male playback, and they don't seem to pay attention to that bat echolocation. But then again, we haven't tried it while they're flying either. But that's likely a good explanation for that second peak in the sensory system. The other thing we found is their sense of vibration was tuned almost a perfect match to the vibrational component of the male's call. So their subgenual organs were tuned right between, you can see this hot spot right in here. So at around 250 to 300 hertz, the female subgenual organ was highly sensitive to that frequency, which happened to be this sensory or the vibrational component of the male's call as it transmits through the ground, not just through the air. All right, what else? So we presume that this modality, the vibrational sensitivity, was probably a predation detection mechanism, right? But that led us to another question about, are the females actually paying attention to that vibration that's being produced by the males, which is an artifact of the male's airborne call as it transmits through the soil. We'll get to that in just a second, too. But there did seem to be a very interesting spectral match between the female's sensory system, subgenual sensory system, and the actual vibration set up by the male's airborne call. All right, so that's some of the things we learned about the females. Then we wanted to find out, what do females really want? It's the big question for all guys, right? What do females really want? And in this case, we really wanted to know, what do female mole crickets really want in a male? And ultimately, how do they deal with all this noisy stuff that goes on in Lex? So the first thing we had to do using basic playback experiments is figure out, what's female choice? Like, is there a female choice? There could be ostensibly no choice, right? Females could just be going to the nearest call. What we found that there were really three things that female prairie mole crickets were paying attention to. One was the loudness of the call. So if there were two choices, no difference in the acoustic quality of the call, she would always go to the louder one, which is a proxy for distance, right? So if you've got two fine male specimens and one is louder than the other, ostensibly the call is lower, like you go to the one that's louder. We also found that female prairie mole crickets really like males that call faster. So the ones that had a higher repetition rate or chirp rate were clearly preferred by females. That's likely a proxy for condition, right? So the male's in good condition, he can forage, he probably has some developmental advantages, maybe some heritable advantages, and so females will always go to the male who chirps fastest, right? The other thing that we were really interested in, though, is females preferred the lower frequency call. So what does that tell us about female choice in terms of mole crickets? Big guys, right? So what they really like, this is really what we really discovered, is they like loud, big, fast chirping fellows, right? And so this is what we learned about female choice. I don't know what that means as far as a human experience, but this is what female prairie mole crickets really like. So these were the larger males that were producing this lower dominant frequency call. So then we wanted to know, okay, so this seems to be putatively like this should be a champion species for dealing with noise, right? These guys have been evolving for the Ensiphera. The crickets have been evolving for 200 million years. This group has been around for at least 100,000 years, mating in this cacophony of sound. So how do females perform in complex cocktail party conditions? And that, again, required now going back to playback experiments. And so what we did then is we know what females prefer. They prefer a lower frequency call to a higher one. So we could synthesize those present the preferred versus non-preferred signal in the context of isomodal noise, right? So this would be acoustic noise that was actually created to resemble the chaotic biotic noise produced in a LEP. Cross-modal noise, that's vibrational noise. So we can actually do playbacks using calibrated shakers through the substrate to create a noisy environment that produces noise through the subgenual system. And then multimodal noise. Throw both of those challenges at the sensory system to females and see if they can still detect the preferred trait in males. And then the other thing we asked about this, what about, given that we saw that females actually are tuned, female sensory system in the the subgenual are tuned to the artifactual vibration of the male's airborne system, does she actually prefer a call that has a vibrational component? In other words, do females prefer a call that's not just unimodal sound, but also multimodal, is tickling the subgenual system as well. Oh, and if you want to see what this looks like, if you scan that with your phone, right, you'll actually see a live playback. I'm not going to show you one right now. Okay, we're going to go quickly. I'm going to wrap up here. So what we found is that in isomodal noise conditions, really loud playing back noise at the same time we're doing playbacks of these preferred and non-preferred traits, females always made the right choice. In noisy conditions, females behaviorally never made a mistake, with the exception of a couple females in trials. But when we introduced cross-modal noise through the substrate that gets transduced through the subgenual organ, all of a sudden females could not tell the difference between a sexy male and a non-preferred male. This is the interesting part of these trials, though, is when we actually coupled cross-modal noise and isomodal noise in a multimodal context, females, for the most part, made the right choice, which is really kind of surprising, because now what we're seeing is a pattern of the sensory system actually essentially using some of the co-modulated properties of the noise to overcome the challenge posed by the cross-modal noise. The other thing we found is that females actually do prefer a male call if it includes a vibrational component too as well, given all things being equal. So this wasn't a preference test. This is two of the same call types, one with vibration in the substrate and one without. But when we introduced overhead noise to that, biotic noise, that preference fell away. The preference for a multimodal type of signal. So going back to the original question that I'm interested in, a lot of other bioacoustics folks, is how does noise modality influence high-stakes decisions in animals? The big thing that we learned from all of this, and thinking about all the dynamics and how the modality of the noise, the modality of the cocktail party noise is really what matters, the modality that matters. When we think about noise effects, like there's a lot of investigators in my field studying anthropogenic noise effects on bird behavior or in marine settings, so there's a lot of work done on that. One of the things that our field is really starting to dial in is what happens when the sensory channel is different than the primary channel that the signaling that's being masked through the cocktail party problem exists. So with that, I'll throw up a whole list of collaborators and students that have worked on these sorts of problems. And again, as I promised, I haven't really given you a lot of answers, but hopefully I've created some questions in your mind about this whole problem of noise and sensory systems and the challenge of organisms in dealing with multimodal, cross-modal, isomodal types of noise in their life histories. And with that, I guess we can take some questions at this point. Do you want me to mute the mic?
Actually, this is the mic right here, right?
Yep.
Why don't we just pass it back and forth? I can read questions off. Oh, I don't need to be videotaped, though. I can read questions off from anybody from the chat rooms that want to chat in, and we'll repeat questions from the room so everybody can hear. We're going to just do questions on chat so we don't get a bunch of overlapping questions coming in at the same time. Can everybody still hear everything? I'm hoping. I had my system crash once, but it's back up. So, oh, good. We're still good. Okay. So, one other thing while questions are going on, anybody who's logged in that has more than one viewer, if you could just type in and tell me how many people are viewing, we'd like to try and keep track of how many people we're reaching. So let's start with questions. There's one here in the room.
So is it possible that the auditory component of the signal is actually just an artifact and vibrational instead of vice versa?
Can you repeat?
Yeah. So Ben asked if the vibrational signal is an artifact of the acoustic or vice versa?
Vice versa.
Vice versa. Okay. So in other words, is maybe the primary information being sent through the substrate and the airborne is just artifactual. That's actually what we think. It's a good question because it's exactly what we think is happening with burying beetles. But we don't think that's what's happening in this system. The males are very clearly, to add some complexity to this problem, the males actually couple their wings to the top of that burrow. So they're actually in contact with the substrate. That's why there's such a robust vibrational signal produced by that. But we can get females to respond in the absence of vibration, and they're very good at responding to just the acoustic call. When I present a call, and we've done this, it's a good question, when we presented a call with no sound and just vibration, they don't respond to it. So they need, it's at least a co-module, it's at least a, they've got to have at least a sound to induce the female response, phonotaxis. So, when?
Could we be dealing with two different cocktail party phenomena here and two different modalities? So step one, find the cocktail party.
Mm-hmm.
And so that's all the guys, kind of the combined loud song that all these guys are doing. And then the second challenge is once the female gets to the cocktail party, she's going to pick somebody, and that's where the sort of the vibration and the signals related to distance might come in.
Mm-hmm.
Right, so a signal that's, because you watch that little video you showed, went right to that track.
She did.
So she must have a system, for, especially at night.
Yeah, where there's no light, right?
Can you repeat the question?
Yeah, so when asked whether or not there's likely a scaled effect here, where females are paying attention to what may be a cocktail party, maybe at a further distance, that's probably a cue, right? That, okay, there's this cacophony of guys, I'm heading that way. Once you get in the system now, you have a variety of signals that are competing with one another, and maybe the vibration actually provides additional information for females